Okay, so we will continue our series of seminars, and today we have the Dr. Alessio Bartocci. So briefly, he got his PhD degree from the University of Perugia in theoretical chemistry. He has an experience as a postdoctoral fellow in different universities from 2016 until 2022. So he has been affiliated to the University of Geneva, l'École Normale Supérieure of Lyon, the University of Trento, and also currently at the University of Strasbourg. His interests are broad also, and part of the research topics focuses on all atom and coarse-grained simulations, ligand parametrization, binding affinities, and kinetic calculations, as well as protein-ligand interactions. And today, well, you can talk about your presentation. So the title is displayed. And welcome, Alessio.
So many thanks, Adolfo, for the introduction and for the opportunity you gave me to show you my last project. And today we'll do a journey through coarse-grained molecular dynamics simulations in order to explain how the cannabinoids could bind in specific receptors as the glycine receptor. So, first of all, I should introduce what branch of chemistry and science study the interaction of such big aggregates. And this one is the supramolecular biochemistry. This is the domain of chemistry that studies the structures, the kinetics and thermodynamics of these large aggregates that are stick together by non-covalent interactions that could lead to reversible aggregation processes like the hydrogen bond, halogen bond, the hydrophobic forces, van der Waals, and electrostatic effects. So my topic will be devoted to the study of protein-ligand interaction in this specific case, but we also could talk about protein-DNA interaction systems and protein-protein systems. So these systems, protein-ligand, are really important because the molecular recognition could refer to a process in which proteins, macromolecules, could interact with these small ligands. There is a high specificity for the protein binding sites and as well for ligand partners. For example, I could introduce chemical physical processes like protein crystallizations that could be mediated and enhanced by ligand binding. In this case, we talk about molecular glue ligands or co-crystallizator. And in other case, ligand could also regulate the protein functions. And this topic is the one I will talk about today. More in detail, I will focus on the pentameric ligand-gated ion channels. They are synaptic receptors that are found mostly in the nervous systems. And they are transmembrane proteins with a pentameric symmetry. And in addition to an extracellular domain where the orthosteric site, where the agonist and antagonist bind, there is also another part that is called transmembrane domain. So the most important function of such receptors are the transmissions of the electric signal in the postsynaptic region. Among all of the agonists that could activate such receptors, there are the GABA, and the receptor class is called in this case the GABA receptor, or the acetylcholine, the serotonin. And more in detail, I will talk about the glycine receptors. So in this case, the agonist is a glycine molecule that when interacts with the receptor, it pushes the protein into an activating form. So in this case, its pore is open and there is an ion flux. More in detail, this form is in equilibrium with other conformational states. In this case, we have also a resting state when, for example, the antagonist binds the pore it's closed and the conformational state is in deactivated and the ions cannot pass by anymore. In addition, these two important states are in equilibrium with desensitized states that can present a different value for the ions current. In addition to all of the molecules that agonists or antagonists that can activate or deactivate the receptor, there are also other molecules that are called allosteric modulators. These allosteric modulators bind in topographical distinct binding sites, more specifically in the transmembrane domain of the protein, and then they could regulate the receptor activity. When I was in the University of Strasbourg under Professor Marco Cecchini, we were working in the allosteric modulation of such receptor, the glycine receptor. They have developed a library that is called GRAL, where all of the molecules that have activity toward the receptor are in. Among all of these molecules, 1/3 targets the transmembrane domain of the receptor, so they are allosteric modulators. Unfortunately, for a lot of them, the binding sites are unknown. So my project, my research was really devoted in finding all of these binding sites for a specific class of molecules. To study such systems, they are quite big. I was saying before to Adolfo that are almost 200,000 atoms because the protein is embedded into the membrane. Then you have all the water and ions and so on. So it could be really complicated to study such system from the quantum point of view. We will need really an atomistic view, of course, but a really long sampling of the conformational space. In such case, are important to study through molecular dynamics simulations. And more in detail, the coarse-grained level resolution could help us by smoothing out the energy landscape and enabling its efficient exploration. So this approach we developed, it can be called the multi-scale approach because it combines coarse-grained molecular dynamic simulations at equilibrium and all-atom molecular dynamic simulations for resolving the structure, the most important structures obtained, and to refine such structures. The two ligands I studied are two cannabinoids, the THC and anandamide. They are two positive allosteric modulators, and it's important to stress out that they are they have a higher affinity toward the active state of the protein. And in literature is well known that they have a modulatory activity toward the receptor. So basically, my approach was to set up the coarse-grained system using the new force field developed by Sosa and co-workers, Martini 3, running molecular dynamic simulations and then analyzing more in detail the most important structures obtained and resolving them at the atomistic level. So, starting by the setup of the system, first of all, it's important to say that the protein structure, the PDB, must be converted into a coarse-grained resolution. In this case, is done using a tool that is called Martinize, and it allows to reduce the complexity of the protein. As you can see, the protein is set up by an extracellular domain and a transmembrane domain, in addition to an intracellular. The transmembrane domain, for the sake of the clarity, was divided into two regions: the upper leaflet part and the lower leaflet part. By talking about the ligands, the first step to parameterize a ligand from the coarse-grained point of view is to map the atomistic structure into beads. In fact, in the coarse-grained, the molecule is represented by beads or interaction centers. They have specific interaction potential as well bonded parameters. So this is done by starting with the mapping. So for all of the atomistic groups that are shown, there are correspective beads type, and then by analyzing, by having as reference atomistic simulations, the bonding distributions of the coarse-grained molecules are obtained. So in parallel, it's important to launch reference atomistic simulations, because they have to be the reference to obtain the parameters. And then, in order to show how the molecule must interact with the environment, the experimental log P between oil and water is calculated at the coarse-grained level and referenced to the experimental one if present. Otherwise, it must be done through the use of atomistic simulations. Here you can see a small video of the system. So what we are looking at are binding and unbinding events. So here I reported the THC into a small part of the simulation. You have a specific concentration of the ligand, in this case THC, that interacts with the protein, and we really focus on the most important binding sites of such interaction. I highlighted the red one randomly, and you can see how it interacts, binds, and then it unbinds, and so on. By analyzing all of the simulations that cover almost 0.5 milliseconds of sampling, you can see that for the two systems, there are very specific spots at the protein surface. In such case, for the THC, you can see these spots in the 2D density maps on the left, and in 3D on the right, both in the upper and the lower leaflet, as well as for anandamide, that we will see it interacts differently with respect to the THC due to of course, to the different chemical structure. How we can, after all of these sampling and statistics, how we can deal with the most important binding events. So in this case, we start analyzing, because we have to filter out all of the non-specific events. So in this case, we start analyzing the binding events. On the right is reported how a binding event is counted. So when the number of contacts with the protein is higher than a cutoff, that is called the cutoff of binding. In this case, our report is 10. It starts, the binding event starts to be counted. And then when it goes down to an unbinding cutoff, the eventing finishes. So in such a way, it's possible to discern between specific bindings and unspecific. So here I've reported the distribution of all of these binding events, and by putting a cutoff on into 100 nanoseconds, we can discern between binding specific and unspecific. So in such a way, the specific binding is longer than the unspecific one, and it could give more information, let's say, to the interaction process. Then, by analyzing all of these specific bindings, we can rank all of the bindings by further analysis into specific binding modes. Here, for example, I've just reported the first 3 more specific binding mode. They count mostly for the more than 10%, let's say the first 2. And then we can obtain information on the residence time of such specific binding modes. So after all of this filter of the simulations, we have we can obtain the most specific structures of the interactions in the binding site. Here I reported the case of the THC. For the THC, you can see there are 2 main interaction sites. One is intersubunit, and it's highlighted in orange. And in this case, the THC molecule is sandwiched. The terpenic core is sandwiched between 2 tryptophan residues, and its tail goes into a small hydrophobic pocket. On the other hand, by analyzing the intrasubunit one, that is the one highlighted in red, the interaction, the protein residues who interact with the THC are mostly, in addition to hydrophobic residues, are also polar, like, because we can see also serine atoms, serine residues in this case. And the interaction conformation of the THC is completely different with respect to the one highlighted in orange. So when we were analyzing such results, a work of Kumar et al. Came out in Nature Communications, where they resolved experimentally the complex glycine receptor THC complex by cryo-EM experiments. And what they said was that they found one specific pocket for the one that is highlighted in cyan in the intrasubunit site, and one that were assumed to be a lipid-like density, but was not as high resolution to be resolved through the intersubunit site, the one highlighted in red. And in this case, this site corresponds to the site we have found in intersubunit, where the THC is interacting mostly with tryptophan molecules. And in the other case, in the intrasubunit, you can see that the experimental pose, that is the one reported in gray, and the obtained pose by our simulations, they are really in a close agreement between themselves. For anandamide, the case shows that there are 3 main binding sites. 2 are intersubunit, the one in orange and the one in red. In such case, the interaction pattern is different, because anandamide, it interacts mostly with its polar head, while the tail could fluctuate in the lipid membrane or interact with hydrophobic residues. And in this case, you have one binding site in the upper leaflet, there is the one in orange, and one binding site in the lower leaflet, that is the one in red. In addition to these 2 binding sites, there is another one that is intrasubunit, the one in green. And in such case, the anandamide penetrates mostly deeply in the receptor chains. This analysis can give also information on which residues are the most implicated in the binding. In fact, here, by doing a contact analysis of all of the trajectories, the most important trajectories, you can see that for the 3 binding sites and binding, let's say, binding modes, there are mostly important residues. For example, in the case of the orange binding mode, there are mostly tryptophan residues, while in the other two cases, serine is very important. For anandamide, it's way similar in interaction. The serine, for example, is really important for the binding in the green one, while in the other cases, also tryptophan could stabilize and hydrophobic residues could stabilize the pose. So to conclude, is this kind of approach that combines coarse-grained molecular dynamic simulations and conversion to all-atom resolutions, allows for detection and understanding of ligand binding sites in complex proteins and environments. For THC, 2 main binding sites were found in agreement with KUM experimental results, and 3 for anandamide. In this case, in the latter, mutation experiments can be tested to prove the protein residues implicated in the binding with mutagenesis experiments by starting from, for example, for the residues we have predicted. In addition, I'd like to stress out that this kind of computational approach is extensible to other functional allosteric proteins like GPCRs, kinases, and to drug design of allosteric modulators and inhibitors. In the end, I'd like to thank Adolfo for giving me again the opportunity to show you my last results. The group of statistical and biological physics in which I'm working right now in the University of Trento, and the group in the University of Strasbourg, where I was under Professor Marco Cecchini. There is one of the authors of this project, as well as the group of molecular biology and structural biochemistry in Lyon, where Paolo Sosa was another one of the authors of the paper, and all of you for your attention.
Thank you, Alessio. Can you hear me, right? So it was a very nice and clear presentation, and now we have quite plenty of time for the discussion. So I will invite anybody who is willing to ask any question just to raise their hand and ask the question. I guess they can unmute themselves also. Okay, there is one question. Yeah, go ahead, Fernando.
Thanks. So very good presentation, very interesting work. I got a question about how easy is to parameterize and validate small molecules in Martini tree.
So with respect to Martini 2, you have to take into account also other parameters. There are the shape and the solvent accessible values of the area of the molecule. So in such case, the trickiest part is to I would say to obtain the non-bonded parameters, because that one gives you the way the molecule interacts with the environment. So the beads and the bead type, they must be chosen carefully. And that one, of course, influences also the bonded parameters. So it's like a try and error procedure. Let's say you start with the topology you have in the atomistic, and then you go refining everything. So, but in general, it will take, for example, in one week, maybe less than 1 week of work. But I can say that the way the ligand are parameterized in Martini 3, they give more detail for the interactions. So it's, if you want to sample such protein ligand interactions, it's the use of also of tiny bits like Martini 3 does, is better to describe also the shape, because you have this kind of ligands, they could also fill pockets, and it's kind of important to have this in mind.
Okay, so did you get the answer, Fernando?
Yeah, thank you very much.
You're welcome.
Okay, so we keep asking more questions to the speaker. So I have one before somebody else runs into. So about the, I mean, you said that your simulations, I guess the millisecond time scale of 1.5 is refers to the complete sampling of all possible. I mean, did you run trajectories, single trajectories in the scale of milliseconds?
No, we have done, you are right, we have done more replicas. Each replica was 10 microseconds. And then in order to obtain 0.5 milliseconds, we run 50 replicas for each system.
So just my question is regarding the time scale in which the protein might, I mean, these binding events could induce. You mentioned that there are some different states in the protein receptor. So can you expect some conformational changes in the, let's say, hundreds of microseconds, or is something that doesn't happen in vivo, let's say?
So what we were expecting is the unbinding of ligands in the term of microseconds. So that's why each replica was run for a long time. But on the other hand, the fact is, when you simulate proteins, and in this case, this big protein, you put elastic networks in order to avoid the conformational changes of the protein. If you want to see conformational changes, in that case, you have to go for the atomistic, but also in that case, you will need a lot of time. In this case, our focus was just to find binding sites. And one of the I mean, something we have in mind is to see if the resting state of the protein, so by starting directly from the resting state, has a different affinity toward the ligand.
Yes, okay. So I got my piece of answer. Okay, I will encourage the people to ask, because it's an opportunity to know a little bit more about Martini 3 in terms of protein and ligand parameterization also. And yeah, there is one question. Yes.
Hi. Really very nice talk. It's very nice to see how the ligand actually finds the binding pocket in the glycine receptor. So the question is that once the ligand has identified its binding pocket, did you thought, or did you try to do this kind of umbrella sampling to estimate the binding free energy or stuff like that? Does it make any effect in understanding the binding pocket? Because you said, like in one of the slide, that there is 3 sites are identified by the ligands.
Yes, it's a very nice question. And I mean, we had in mind to perform umbrella sampling, but we were also lucky that in such case you have sampling at equilibrium. So all of the probabilities you have for binding and unbinding can give you directly the delta G for the pocket, in that case for the site. And then if you want to obtain a standard delta G, you have to correct for the volume and for the concentration. But in order to see the delta G, you have already the values. So all the probabilities that you saw, for example, in the plot of the decay, in that case, you have directly the binding and unbinding probabilities. So, but in general, you can also perform the umbrella sampling. You start from the most important structure, in that case, the one we have obtained, and you go for the detachment of the ligand, and in principle, it should have a very similar result with respect to the one you obtain from just sampling.
Thank you very much.
You're welcome.
All right. So we still have some time. Okay, I have one just last, I promise, questions regarding the fact about the contact analysis.
Yes.
Between the ligand and the protein. So is this just a cutoff base analysis, or did you perform some specific chemistry-based contact analysis?
So in this case, we just used like we chose the cutoff because from other simulations we saw that after 5 contacts the molecule is not interacting much, so it could give rise to a specific event. In principle, you can put like the minimum of the cutoff, for example 1. In that case, you will have all of the sampling. But what we did was just to filter out all of the non-specific parts. For example, in one binding event, if you had put like the cutoff to 1, for example, the unbinding cutoff, you would have many more frames that maybe could lead to unspecific events, or maybe the ligand could also migrate toward other sites that are very close to the binding pocket. In such a way, you are sure that the site remains that one, so you are not sampling other like closer sites.
Mm-hmm. Yeah.
But in principle, yes, you can also tune the cutoff. Of course, you can do, for example, like some calculate in order to see the binding events by changing the unbinding cutoff to see if the number of contacts and of events changes so much, and then you will be, if you are a convergence, in principle, you should be lucky.
Mm-hmm. So because specifically the cutoff that you use here, it's a small, yeah? Because
yes.
20 contacts, you mean a specific between atom, right?
Yeah, between bead and bead, yes.
Bead and bead, yeah.
Yes, for sure.
Yeah,
between 2 particles, yeah.
Mm-hmm. Mm-hmm. Okay. Mm-hmm. All right. So now is time to say thank you. You have many. There is one more. Yeah.
Okay, one more. Is it possible to use this approach to uncover cryptic pockets?
Yes, I mean, you mean cryptic pockets, something that is already formed, or it forms after the, for example, an allosteric pathway?
Yeah, that these kind of pockets that just exist for a brief period or under some circumstances. Yeah.
Okay, so in principle, if you, for example, if you obtain for all atom simulations a cryptic pocket, then you can convert the structure, the protein structure is coarse-grained, you put the elastic network, and in principle, the cryptic pocket should be there for for the entire coarse-grained simulations. And with this approach, you should be able to sample the cryptic pocket. But the problem is that if you don't want the cryptic pocket to be to have this long lifetime in such way, maybe the only way is to use atomistic simulations. But if you want to sample for a very long lifetime, it's possible. Yeah, you have to freeze the protein and then you do sample.
Ah, okay. Oh, thank you. Well, doing this in all atom simulation is pretty expensive, actually.
Yes, that's why, because it's mostly, I would say that is just an idea. This one, you freeze the protein into a specific state, and then you sample that specific state with the ligands. Of course, if you compare that state with, for example, with the initial one, you will obtain a different delta G, so different binding events. So in principle, yeah, you should go for it.
Okay. Okay, thank you.
You're welcome.
I guess also Fernando's question goes around as you said, the elastic network is a very robust model if you want to retain the conformation of the protein, right? But if it has to undergo even some, let's say, like in this case, uncover some part of the protein which before it was hidden because there was not, let's say, the allosteric communication was not yet reached till that moment. So then you need something like to tune that, or either to use the equivalent which we develop and we are working also is the Go Martini, where you can also impose these flexible bonds equivalent to the elastic network, but with more, let's say, Lennard-Jones like behavior, and then perhaps the protein could uncover. We could test, right? So if we have a protein model, maybe we could test and search for this. Maybe it's one way to go.
Yes.
All right. Okay. So I mean, I have to say thank you because it was a very interesting presentation, Alessio, and hopefully we stay in touch. And in 2 weeks we have another seminar, and I invite all of you to participate again.
Thanks.
Have a nice afternoon. Okay.
You too. Thanks a lot to everyone. See you. Many thanks.
Bye bye.
Yeah.